Thank you everyone for being part of this awesome event, and thank you so much for coming. My name is Ahmed. So I'm a postdoc with Ross, and I've been with Ross for actually over 5 years. I'm going to tell you a little bit about what I do. But really, what I want to talk about is the concept of flies and how we can move from you designing your questions and even answering them, with flies. So I'm going to use, cancer as an example, and I'm actually going to allude to the RET model that was also mentioned and some of the things that we do in the room. Right, so I'm going to follow this outline. So I'm going to introduce you to what I do, talk to you about just the Drosophila genome, and also actually the beauty of Drosophila genetics regarding the naming system in flies, because that's very important, when you're actually working with flies. Tell you a little bit about development in Drosophila, the tools that we've got in Drosophila, and also introduction to crosses. And I'm going to show you some of the online tools, online resources that we've got in the fly world that can help what you guys are doing. Right. So what I'm doing in the lab, disease and drug, compounds maybe. So what I do in the lab is actually I'm moving from patients into developing models to model this patient and using various techniques to actually study the models. And the goal of that is to bring about new therapeutics, repurposing existing therapeutics and taking that back to patient. So that's really the summary of what I do. We can talk in detail about the detailed methodology, at some point during the workshop. And I think Ross has, done a very good job, So these are organoids. So I'm working with organoids. So organoids are different from assembloids. I'm also working with assembloids, but that's another level. So for now, the data I've generated are organoids. So I'm looking to also use assemblers at some point. Is that okay? So, and also I'm developing, new tools in organoids. So, I'm an organoid expert, not yet an assembloid expert. Is that okay? Right. So I'm developing models, test using some tools to test some of my questions on them, and then trying to, to move drugs to patients. Right. So, but I can tell you about the details later. So, right. So stop, not stop. Right. So the Drosophila genome. So in Drosophila, we've got 4 chromosomes, right? So we've got the X chromosome for female, X over X chromosome. We've got the second chromosome, we've got the third chromosome, and then we've got the fourth chromosome. And in Drosophila, and that's the female, sorry. And then in the male, we've got the XY chromosome, and then you've got the other chromosomes. And in Drosophila, similar to in human, the genes are annotated onto the chromosome. And that is why we use Drosophila mainly for genetics, disease or genetic study. Is that okay? So because we've mapped, so in the past, people have actually mapped, the different locus on the chromosome, and you can begin to go individually into these genes and ask, oh, let's say if I perturb this gene, what happens? Let's say I insert something into a particular locus in the fly, what happened? Is that okay? And from the database and what people have actually, we know that there were about 17,000 genes in flies. Is that okay? And the power of what we've got these days that for example, people that started working with flies, let's say 50 years ago, don't have is we've got database that we can easily go into and we can ask, what are those genes that are just on the X chromosomes? What are those genes? What are those genes that are just on the second chromosome? What are those genes that are just on the 2L and the 2R? So, and this you're going to understand better why we're depicting the 2L and the 2R later when I talk about what we call recombination. Is that okay? So we've got 2 homologous chromosomes. We've got 2 arms of the chromosomes, and they're really useful for Drosophila genetics. Is that okay? So in addition to our database, that database allows us to know the exact location of a particular gene, and we can manipulate that gene in that location. So now I know you guys have heard of technique called CRISPR-Cas9, right? So that allows you to edit things, in the genome. And without annotating the cytological position of genes in the genome, you won't be able to do that. So in flies, we know the position of things as well, and we can easily modulate those genes in that position, right? Right. So the next thing I'm going to show you, and the reason why I needed the board is to write something about what I'm going to be showing you. Right. So when you work with flies compared to people that work with cell lines, so with cell lines, what you need to write when you're reporting your, let's say, what your data is, oh, the name of the cell line. In some conditions, or in some cases, sorry, you can include, oh, this gene actually I've got GFP, for example. Sorry, this cells I've got GFP. For example, oh, I've overexpressed KRAS in this cell line. Is that okay? But in Drosophila, we've got, a particular way of actually writing out the genotype of the flies. And in mouse as well, people have got, the way of writing out, the genotypes, of the animal. But in flies, we take that really seriously, because we know that once you write the wrong genotype, that will give us wrong information about your strain. And also, people that want to use that strain, that is going to be useless for them. So this is a very key skill to have, when you're actually working with flies. Because, for example, if you've got like red-eyed flies, they're all looking the same. So, but if you can annotate that genotype is KRAS, this genotype is, for example, APC, that would be a problem. So one way to actually name things in fly is that we use descriptive phenotype. So what do I mean by that? So basically, flies that are white-eyed, you can annotate them by white. Is that okay? So flies that have got the curly wings, you can be like, they're curly. Okay. So, and then flies that can transform from, let's say you induce what we call a transformer gene in flies. So the transformer genes actually depict what goes to male or female. So you can actually induce a male, let's say, gut in a female animal, and people are doing that. Is that okay? So, and that's a transformer gene. So we can use the phenotype to name, the fly that we're working with. Is that okay? So the next thing about the naming is that we represent recessive genes. So let's say, for example, you need 2 copies of a gene to see the phenotype, right? So, and that's recessive. So those are represented by lowercase. So for example, the white-eyed phenotype. Is that okay? So the white-eyed phenotype, because it is, you need 2 copies of white-eyed to really say that. So it is represented as lowercase. So we call that white minus. Okay? For example, notch is a dominant gene. So we use capital N for notch. Is that okay? So for example, curly is, what depicts curly is actually dominant as well. So we write that as capital C and then Y. So yeah, so recessive genes are, they're labeled as, as lowercase and dominant uppercase, right? So that's very important in flies. Right, so genotypes are written according to the linkage order. So now when you look at some fly strain, people will write, let's say, YW. Is that okay? So not WY. Is that okay? So the reason for that is that on the first chromosome, actually the yellow gene comes before the white gene. So then you need to write the Y first. So because they're on the same chromosome, but you need to annotate that this comes first on the chromosome, and then this comes second, this comes third. So that's how you write your genotypes in flies. So you can't write it the other way around. Okay. And that is very important, because you know that, oh yeah, so the first thing that is, let's say, mutated in this, the first phenotype here is white, is yellow, and then the other one is white. So let's say, for example, then you've created your own transgene, and then you know that, oh, actually, in this position, I've got, let's say on the 20L position, for example, you've got KRAS inserted in it. Is that okay? And then let's say on the 40L position, you've got, let's say, APC inserted in it. So you have to write KRAS first before you write APC. So not because A comes before K, but because KRAS actually is inserted on a location that comes before APC. So yeah, so the location of genes actually matter, and you write them in the order, right? So also, symbols of genes on different homologous chromosomes also on different homologs of the same chromosome are separated by solidus or dash. So basically, let's say for example, you've got white-eyed animal, for example. So white-eyed animal would have W over W. Is that okay? So these are white-eyed animal, and we'll call them they will have what we call the white minus. Is that okay? So because they are homologs of the same chromosome, you separate them by what we call, you can do dash, or you can use solidus. Is that okay? So let's say, for example, you're thinking of, let's say you want to create a transgene. We know that sometimes, you know, the white, there can be a wild type chromosome homologs on that on like in your fly basically. So what you can do, you can write something that I'm like, this is white minus over plus. Is that okay? So the second chromosome is wild type for white, but the other one is mutant for white. Okay. And I'll tell you what the phenotype will look like later. So what you can also do is, let's say for example, you've actually inserted, I'll use KRAS because KRAS is quite popular, right? So let's say you've inserted KRAS onto one of your chromosomes, right? So ideally the way you actually write KRAS as an homozygous is, you've got KRAS over KRAS, right? So let's say it is heterozygous, you can have, and then it is balanced, right, for some reason. So you can have, KRAS over your balancer. So now, who will be able to tell me what chromosome is this? So KRAS over CYO. Because I think we sent you some material and I've given you some of the, clue actually. So who can tell me what chromosome, KRAS is inserted in? So because it's over CYO. So any answer? No. So that is inserted on the second chromosome because we know that, so the curly balancer, so curly is actually a form of balancer. I'm going to show you what balancers are later. So for example, someone that works with flies, I know that, because this is over curly, meaning that KRAS is inserted on the second chromosome. So you can now appreciate that naming your flies properly matters. So let's say, for example, you wrote KRAS over KRAS. The first question I'm going to ask is, so where is KRAS inserted? Okay? Because I need that to be able to do other experiments. Okay. So yeah, so
certain positions in chromosome that can be calling what's please.
Yeah, so because these are actual, so Kali is one of what we call balancers. So these are balancers. And also what we call a mark. Is that okay? So the Kali balancer itself is inserted just on the second chromosome. Is that okay? So and then it gives you that curly wings phenotype. So it's always going to be there. So you can create your own curly phenotype in a different region of the animal. Maybe you can have it in the third chromosome if you want. But so far, the only curly that we use is on the second chromosome. And also we've got different types of balancer that also have curly phenotype. Is that okay? And we've got one in the lab that is a co-joint balancer that co-joins the second and the third. That also have curly phenotype, but that's a co-joint balance. Is that okay? So and I'll show you that later. Right. So gene symbols on different chromosomes are actually separated by semicolon. Is that okay? So let's say, for example, you've got a fly that is white-eyed, and you've inserted KRAS into the second chromosome of that fly. Is that okay? I'll show you how you're going to insert KRAS. So now, the way you're going to write that genotype is that it is white-eyed, remember. So white-eyed needs to be this. So white-eyed animal, they're homozygous for the white minus mutation. And then you've got your semicolon, and then you've got KRAS. And because I want to insert KRAS on the second chromosome, I'd love to use curly balancing. Good. So curly balancing. Is that okay? So you can see the homologs are separated by dash. The chromosomes are separated by C. So you need to be able to write that when you're reporting maybe your experiments or you're trying to publish your paper. Okay. And also, even this is very important for others that might want to use the flies that you've used, because they know that, oh, this is the genotype of that fly. All right, so the next thing is that, so the absence of an homologous chromosome implies homozygous. So let's say, for example, someone said, you know what, rather than writing this, white minus over white minus, another way to write it is white minus semicolon KRAS over curly. Okay. So because you know that your animal is homozygous for white minus, so you don't need to write it twice. So, but the reason why we have to write KRAS over curly is because KRAS homolog is different from curly. Is that okay? So, and that is why we need to write that. And do you know that because you are inserting KRAS into this system, those items that your animal are not going to be white minus. So do you know the reason for that? So I'm going to allude to this concept of what we call P elements later. Is that okay? So those items that once you insert something into the animal, they're not going to be white minus. They can have what we call a mini white gene, but they won't be white minus. But I'll allude to that. But for the concept of naming, we can have something like that. Right. So yeah, so uppercase represent protein products, so and without italics. So yeah, so that is self-explanatory, right? So that is just for you to, let's say you're trying to report something in your paper and you're referring to the protein, form of your gene, you have to write that, without italics. Especially in flies. And in mouse as well, they've got their own way of. Right. So in these days, actually, allele identification, carry the number. Is that okay? And these numbers are often in superscript. And in the absence of a superscript, you know that is the first allele that was identified. So in those days, what people do is they do what we call, forward genetic screening. So what they can do is they can add mutagens to fly food, feed that to the flies, and isolate, the mutations that come off that. Yeah. So a lot of them actually, they're like, oh, the first thing that we identify, for example, notch 1, notch 2. Is that okay? So for example, EGFR1. Is that okay? So, and in some cases, some people also use the date that it was isolated. Okay. So, but for us to be able to understand it easily, I think numbering system works better because you can know that, oh, this was the first, allele that was identified for this particular gene. This was the second, and this was the third. So I work with a gene called nonstop. So, and the first allele that was identified was not one, basically. And the second one was something random. So, but the goal here is for you to be able to annotate and name stuff properly, so that other people can easily go into it and use it. And the reason why that was so these days, this is not very important because you can easily go onto database and even know when these things were identified. But in those days, for example, maybe when Ross was doing his PhD or undergrad, you have to go into this lab and ask, can I get this flies? When was it isolated? And people can go back to their lab books and be like, oh, it was isolated in 1978. And also that goes into the numbering system or the naming system of the flies. That okay? So, but these days we actually don't, we don't really use it. Right. So, and so confuse a lot of people and confuse me as well when I started working with flies. Is that wait, so plus actually denotes wild type allele, and minus, denotes a complete deletion of that gene of interest. Is that okay? So when you see people writing white plus, so what do you think the eye color? Because so now the white is referring to the eye color. So what do you think the eye color of this is going to be? It's going to be red, right? But I thought it should be white because it says white plus. So why is it red? Good. Is that okay? So, and then we've got what we call white minus, right? So, and what's the eye color of white minus? Good. Is that okay? So white minus, because it's telling you that what codes for the for the eye color that is red is not there. Is that okay? So, and that refers to as, and then you denote that as minus because it's a loss of that particular gene. There can be point mutation in your gene of interest as well. So remember, I said this is a complete deletion, right? So there can be point mutation as well, right? So also people denote that, depends on what, some people denote that as minus because sometimes a loss of function mutation. Is that okay? And sometimes it can be a gain of function mutation as well. Exactly. And some people, rather than denoting it as minus or plus, people will actually go in directly and write it out. For example, there was a gene called Yorkie, and there was a mutant of Yorkie called serine to alanine. Is that okay? There's a point mutation of serine to alanine. So people can write it that way. Is that okay? So, and you don't need to write whether it's Yorkie plus or minus. Makes sense. So yeah, so and if you don't have plus, that means it's wild type. So if you don't write plus, so for example, if I'm sometimes people will write just white, just this doubling without the minus. So that denotes wild type as well. Makes sense. Right. So sometimes we also use description names for alleles. So in the lab and in DRTC, you guys will see something called bin TS. So I won't go into detail of bin TS yet. So bin TS, is that okay? So bin is a GAL4 driver, but the reason why it is TS is because it is temperature sensitive. And we're going to explain what that means. Is that okay? So, and you're going to use that, I think, in one of your projects as well. So, and that is a GAL4 driver that we use in colorectal cancer, especially to express genes or to drive genes that are required in CRC. Is that okay? So, temperature sensitive, sensitivity allows you to calibrate things. So once you put your experiment at a low temperature, for example, something might happen. Depends on what the temperature sensitivity is doing. Is that okay? And in some cases, once you put at high temperature, that thing is not there. Is that okay? So something that is really sensitive to temperature, change in temperature. So at low temperature, you can have activity or not. Depends on what system you're working with. And at high temperature, on or off, basically. So yeah, and then we've got recessive, we've got not real symptoms. Right. So so the one of the biggest thing in flies is that we always generate transformants. So no matter what you want to do, you want to work with you want to work on colorectal cancer, you want to work on breast cancer, you want to work on metabolic disease, you need to generate transformants. There are people that are working with there are people that are working with just the wild type animal, without generating transformers, but there is limit to what they can do. Okay. So transformers allow us to actually go deeper into questions. And what we can, for example, you want to create, let's say, apoptosis sensor, for example. You can't just create that with you can't just study that with wild type animal. So you can create your own sensor, and that will allow you to study. And during this stage in development, this is what apoptosis does. A necrotic sense, you need a transformer to do it. So, and I'll show you how people can do that. Okay. So now, because transformers are very important, so you need to know how to name them. So transformers names include the p element vector, so the vector that you've used in the inserted genes that you've used in a square bracket, okay? And the location of that thing in parentheses. So, for example, here the full genotype of the short form of this guy is actually UASGFP. So in the lab, I'll be like, oh, UASGFP on the third, for example. Is that okay? Because of that location. So 68, So I think so the chromosome 3 start from 60 to 80. So, and I think chromosomes, I show you in the previous slide. I actually can't remember. But definitely 68 is chromosome 3. So you can write, so you can include the cytological position in this bracket, and then the P element itself is in square bracket. So anyone that sees that knows that this GFP is got yellow, so it's going to be yellow body. It's going to be red-eyed. White is actually mainly used for white agent, so it can be yellow eye, sometimes orange eye, sometimes red. So yeah, and then it's got 10 times UAS membrane tagged GFP. Okay, and then the location is there, 68A4. So another way to write this is you can put everything in this bracket, and then you can write what we call at P2. So now these days, rather than, so in those days, you can insert things in cytological position yourself, but now we've got companies that actually can do that for you. So they've got strains that will allow you to direct your insertion to a particular site using a little bit of molecular biology trick. Is that okay? So yeah, so here, the third chromosome is also denoted by the ATP2 site. And what you can then do is, let's say you've got a different insertion on the third chromosome, and that is, let's say if this is on the right arm of the third chromosome, and there is something on the left arm of the third chromosome, what you can do is you can then recombine them together to have, you can have UAS at P2, and maybe, USGFP at P2, and UAS RFP, something else on the third as well, because you've recombined them. And knowing this location is very important for what we call the combination experiment in flight, because you can't recombine something that is on the second chromosome with something that is on the third chromosome. Although you can combine them, using your genetic trick or crossing scheme, but recombination itself takes place, with things that are on different arms of the chromosome, right? Right. And my take-home message is that, never trust the stock label. And make sure you find a way to, to verify them. Is that okay? Anytime you can. And what are the ways to verify them? Good. You said how, right? Good question. So, right. So let's say, for example, I gave you a fly strain. And then I told you that, what? This fly strain has gone, let's say, I love exokinase for some reason. So exokinase on the third chromosome, it is yellow body, it is red-eyed, it has got curly. But you know what, my exokinase, UAS exokinase, I inserted it on the second chromosome. So how would you verify that? So one thing you can do is you can look at the animal itself. Oh, I said it's yellow body. Are they actually yellow? You'll be able to tell that under the microscope. Is that okay? Oh, I see the red eye. Are they actually red eye? You'll be able to tell that on the microscope. The curly, are they actually curly? You'll be able to test that on the microscope. Now let's go to things that you can't actually tell apart. You can't see. So now you need molecular trick to do that. So for example, I said you've got exokinase. So you can PCR exokinase out and see, oh, do I see exokinase in this thing? You can sequence it and ask, oh, is exokinase still there? Okay. So because you know the size of what you're looking for, so you can do things like that. You can even blot it and add antibody and ask. You can image it. You can ask, oh, I've got anti-exokinase. Can I pull that out in my stream? So we always try to make sure that we're working with no, we must work with the right flies. That there was no bargaining about that. But we always try and verify things before we actually bet on it. Is that okay? Especially when they're not generated in our lab. Because there were stories of people that, for example, worked with the wrong plasmid, and they had to retract the paper. The paper was published in, I think, Cell or Development, so very top journal. So and they had to retract the paper because the person used a different plasmid to create their transformants. Is that okay? So and you have to, because it's a lie, so you have to retract it. So these things are very, very important, and they're key to Drosophila research. Is that okay? Right. So now, Drosophila and development. So I think Fumlayo already, showed you, the different life cycle, that we've got in flies. So the start from embryo, you've got the larva stage, you've got the pre-pupa stage, you've got the adults, and from the pre-pupa, you've got the pupa stage, and we've got the adults, right? So, and I'm happy to tell you that every stage of this organism is actually important for research. And you can, so the thing is, so people that actually develop the Drosophila research, you know, people started, let's say, from one lab, for example, but everyone actually took out something to start their own thing, right? So, and then from there, others also took out. So, because we are talking about colorectal cancer here, it doesn't mean you should start studying colorectal cancer. So, the goal of this is to think about, wait, what part of this animal can I use for what I'm interested in? What stage of this animal can I use for what I'm interested in? Because all the stages are unique for what you can use them for. Is that okay? So let's say, for example, you're looking at, the effect of, toxins on something. What you can do is, even you can just add the toxins to the food. You can collect the embryo without them even feeding, just for you to look at that effect directly. And once you collect embryo, there are ways to collect it. So yeah, so you can use any stage or of the animal to actually study things that you're interested in. I think once they're relevant, it's like, and every stage have got their own positive and negatives as well. So right, so and then during the development of the fly, they are sensitive to temperature. Is that okay? So the life cycle from egg to adults at 12 degrees actually take about 50 days. At 18 degrees takes about 19 days. At 28 degrees takes about 7 days. Okay. So they're temperature sensitive as well. And I think the reason for that, someone is asking that, because for example, people in Nigeria live in, let's say, high temperature, is that going to affect the life expectancy as well? I actually don't know. That might be the reason. But temperature affects temperature, temperature affects, the Drosophila life cycle and their development. Right. The beauty of Drosophila is that, like Ross alluded to, that we've got similar anatomical structure. So I won't say similar, but they've got representative of the anatomical structure that are in human. So for example, they've got digestive systems. They've got nice body organization that you can use to study, anterior, posterior polarity, organization of the cells, the epithelium and things like that. The circulatory system as well. Okay. So blood, they don't have red blood cells, they don't have, white blood cells, but they've got immune cells. So they can be used to study some of these good biology questions, because they've got similar anatomical structure compared to human. And the beauty of Drosophila is also, like I mentioned earlier on, you can use embryo to study some question, lab to study some question, and adults to study some question. In addition, the tissue in the animal. So Ross is known to have pioneered the eye development. So he is a very good dissector. It can dissect like the eye disc, maybe 1 in 2 seconds, like can dissect a lot. So yeah, and I love to dissect the gut. Is that okay? So I can dissect maybe one gut for like let's say 2 minutes, right? So and you can dissect, one eye disc in 2 seconds. So right, so people work with different part of this, and there are people that like lots of labs working on the wing disc. So using it to ask questions regarding apoptosis, the effect of radiation, on development, on even polarity of cells, how cells divide, the effect of, for example, toxins on things, on this type of things. That's something that you can look at, proliferation. So people look at that. So also the genitalia. So people that want to look at maybe prostate cancer, this could be something that you might want to look at. So there are people that look at that use the gut. So the mid gut, the hind gut, the foregut, people use that. And a lot of people use the eye. So the eye disc develops into the eye. So people use the eye for screening, for developmental biology study, for cell biology study, for basic, cell biology questions as well. So people use almost every part of the animal. The wing, the wing disc will develop into the wing. So, people use the adult wing to study the research question. And the beauty is that so there was a lab in Sheffield, so that actually they're very big on the planar polarity, as a system, and they've developed, they're actually looking at the membrane. So they've got, receptors, ligands that interact, and using, this ring disc as a system, they've developed not only from cell biology but to computational biology. And the beauty of what they are doing is they've actually translated this as well. Exactly. So, it's a very, very, very big lab. I keep blanking on his name, but a very popular lab in England. So, these are people that, not everyone is going to work with the guts. That's just my, that's what I'm trying to do. Right. Now, one of the bread to the butter of, of Drosophila research are the markers and the balancer, right? So in a Drosophila research lab, you want to be able to tell the differences between all these. Note that in your lab, you might not use all of this, but you're going to use some of them. Is that okay? So being able to tell what a wild type would look like, forget about the cyan colored ring. That's just Andres doing his own art thing. So forget about that. So, but there are some animals that are going to be, wild type for the eye color. There are some for the body color, and there are some that are going to be, what we call the ebony phenotype, and there are some that are going to be yellow bodied. Okay, there are white eyed. There are some that are going to be the mini whites. Is that okay? Because of the insertion of the P element. There are some that are going to be bad eye, so the bin eye shape. There are some that are going to be drop, and there are some that have got the irregular facet. So note that the balancers are quite different from markers. So what we use balancers for is to prevent recombination of the homologous chromosome. So for example, let's say you've got KRAS as an insertion over, let's say, exokinase as an insertion. Generally, it is known that in the female animal, there's going to be recombination between these two, homologous chromosomes. So for example, let me write that out. So let's say you've got KRAS over exo, I just do X. Is that okay? So UAS KRAS. Sorry about my writing. UASX, for example. So in female animal, there's going to be recombination. So which means, some of your animals even might lack KRAS. But to prevent that, we always use balancers. So, and that is why you can see me writing, UAS KRAS over, let's say, for example, curly. Is that okay? So because no recombination will happen here. So anyway, so the only thing you can get is if KRAS is homozygous viable. So you can have KRAS over KRAS, that's fine. We're happy with that. But you can't have, curly over curly because that will kill the animal because that's a rule. That's how balancer works. So, but that prevents recombination. That's just the concept of balancer. But the markers are not necessarily balancer. So the markers allows you to track your insertion, allows you to track chromosome. For example, irregular facet is not a balancer, it's actually a marker. Okay. So that is why when we are playing with Drosophila crossing schemes, we always try and, so in animals that have got irregular facet, we always try and make sure that they're male. And the reason why they need to be male is because recombination don't take place in male. That's a rule. Exactly. So this is, for example, a marker, not a balancer. But like mini white is also a marker, not a balancer. But I'll show you some of the, some of the balances. Like for example, curly is a balancer, is also a marker. So it's called the curly ring, but you can use it as a balancer, in the experiment. Serrate So that type of wing, they've got broken wing towards the tail. So, and then we've got what we call very short wing, and we've got a like tail. So compared to the wild type, is lacking that. So we don't generally use this in the lab. I use this in my PhD. I've actually not used it of recent. Curly is the most popular. We use it every time. We've got this. We use this every time. I've used bad guy actually. Yeah. So you don't need to use everything. You just need some to run it. And then for the bristles, we've got emerald. We use that a lot. Is that okay? And even DRTC, I've got that, and I think you see that as well. I've never used pin, So by the way, Humera is a marker, but the balancer is called TM6B. So there is a balancer called TM6B that's got the Humera marker, and that's what we use in experiment. Also, serrate is often tagged with a balancer called TM3. So TM3 serrate. So and that's what we use. We use stubble a lot as well. So stubble is TM3 stubble. Use that. I've never used the singe phenotype. So so these are phenotypes that can be tagged with balancer. So here actually, these are markers, and the only balancer here to me is curly. Is that okay? So every other thing here can also be tagged with the balancer so that you can use that for experiment. Right. Now, I've showed you the adult balance. So there are some balances that are useful for you to select your larva, even some for embryo. So we've got someone in the lab that he is trying to separate, GFP positive embryo from negative now. So, that we've got things that can allow you to do. So, but for example, the black cells, in lab allows you to separate wild type, from the mutant or from what you're interested in. So let's say, for example, you've got something on this chromosome, on the black cell chromosome, you can select just them rather than selecting both, wild type and this. Also, Tobin marker, really useful. So this is, I dream about this every day because I use this a lot in my experiment. So, so the Tobin marker, quite useful, long is normal, short is Tobin. And now you can separate male from female, pupa, even at larval stage, because the genitals here is actually transparent compared to the females that have got, lots of fat surrounding this. So we can separate male and female, sorry, female and male, larva, pupa, and even you can use that for your virgin collection and be like, I can separate this, so I'll use it. Right, so some of the technology in flies, and one thing that flies are really good for is called the classical forward genetic screening. So what people do those days is that, and I'm sure Ross did this as well, is that it actually fed, this is by the way a carcinogen, just so you know. So so he actually fed EMS to flies, and trying to isolate various mutants from them. Is that okay? So in those days, you feed your male animal with the carcinogen. You cross them with wild type female animals, and then you will cross them again. The offspring might carry the mutation. Even if they don't, you can select for the right thing later. So it's just a little bit of crossing, not even a little bit, like few generation of crosses, to identify, the mutants that you're interested in. So ideally, what you can do is people will start with the white-eyed animal or even the red-eyed animal, but so you're going to be picking individual animals. Is that okay? And you're going to be figuring out if that animal is actually is carrying your mutant. Some of them won't, some of them will. And you can, with the power of, plotting systems and things like that, you can figure out, what happens to the mutation, what mutation have I got. Now we are lucky because you can sequence them. So, you can use sequencing to be like, this population of animals, what mutations have we got with them? Because they should all have that mutation because design your crosses to generate that. But lucky us, we don't need to do that anymore. So now we've got tools to actually probe individual genes in the Drosophila genome. And if we don't have that, you can easily design yours. So designing that, there were people that would, we've got a database for you to go on, look at the RNAi that you're interested in, and then people that can short that into flies for you. So this 4 genetic screening, the classical using EMS, because EMS is carcinogenic. I don't think we can use that in the lab anymore. So yeah, no. So we can't use that in the lab anymore. So it's been replaced by what we call the RNAi experiments, the CRISPR screen. Now, so what RNAi does is that you've got your own gene of interest. So you can design what we call hairpins that will target that thing and knock it down, reduce the level basically. So preventing it from being translated. Correct. That is the same principle, yeah. So, but in flies, remember, we said you've got your hairpins, right, that targets your gene. So basically, those hairpins needs to be under the regulation of UAS. So I'll show you that. So UAS is one of the bread to the butter of Drosophila research as well. So and I'll show you what that. Yeah.
So for once they are released itself,
correct. Correct.
Well, feel like now that's same one.
Okay.
Are you going to design the I can tell you, or you have specific images?
Thank you so much. Right. So let's say, so what gene are you interested in? Good. So there are buckets of P53, RNAi out there for flies. So, and how would you know them? So basically, we've got database that stores these things, and we've got stock centers that distribute these things. Is that okay? So basically, you can go online to, one of them is called the Bloomington Drosophila Stock Center. Is that okay? I'll show you some of these. Yeah. So, and then you can go online and so we've got Flybase, that's one thing. And then we've got the stock center. So from Flybase, you can insert P53 and it's going to tell you variety of reagents that are available. Exactly. So, and then you'll be able to tell whether it's from knockdown or not once you get into the list. Is that okay? So the database haven't got the ability to separate, RNAi from overexpression yet. Everything is the same. But as a scientist, you will be able to tell, oh, that's an RNAi strain. Oh, this is for overexpression. And that's something that we can figure out together. But once you go from Flybase, you can go on to Bloomington Drosophila Stock Center. You're going to get a list of RNAi from there. So on Flybase as well, you're going to get lists from VDRC. So this is the Vienna Drosophila Stock Center. You're also going to, so actually VDRC was developed or was created for RNAi reagents. So unlike Bloomington that has got a mix of everything that we can allude to later, but VDRC was actually developed for RNA. Now, they've also to their own, because it's difficult to maintain this, so they're maintaining hundred thousands of stocks, right? So to actually maintain this, it takes, a lot of things that I can't be talking about right now. So sometimes they have to, the stock center needs to talk together and be like, oh, wait, do you have this or we don't have that? And they can shuttle things down, or reduce what they've got. So now VDRC also have got CRISPR reagents. Is that okay? So that allows you to do there was something called CRISPR off and CRISPR on. CRISPR inactivation and CRISPR activation. So they've got reagents, they got the RNA that will allow you to activate the gene of interest and also guide the RNA that will allow it to shut down. So we've got centers that have got it. So you just need to know your gene of. So the next thing is then creating, your own transgene. So, transformation in Drosophila. And that was developed by Ross's friends. I know you know them very well. So yeah, so actually it was developed by Gerard Rubin and Alan Stradlin. I mean, people in the lab and also them as well. So, and you can see, that's developed a while ago. So That changes everything. So in the past, and that I'm not aware of, and Ross will know better. Actually keeping your stock of, let's say, forward genetic strain and trying to do that is actually difficult. But being able to actually transform flies, like you can transform your cells, can transform, let's say, bacteria, that changes everything. And that is the basis of Drosophila transgenesis. So one thing that, and then I'm going to show you how that has been done. So the concept of that is that you've got something called an enzyme called transposase that can cut DNA and allows you to just inject or insert something into it. Is that okay? So, and then you need a little bit of molecular biology to then design your own P element. So what do you want to have in your P element? Do you want to have a GFP there? Do you want to have KRAS mutant? So let's say, for example, you go into the database and you found that actually KRAS G12V is very, very popular in colorectal cancer patients, or is recurrent in colorectal cancer patients. Can I then sequence that or generate that and shuttle that into my P element? So, and that can replace your Laxate. So let's say you're trying to target something and you found that, oh, this RNA or this epin will target the gene. You can shuttle that and replace that with the Laxate. Yeah. So you will need a plasmid, but you need a little bit of molecular biology, to cut and paste. She's the guru of molecular biology. So yeah, so cut and paste, so that's the concept. So you can cut and paste anything, even human genes, you can cut them and paste them into your P element. Yeah. Yeah. Yeah.
So specific.
Very good. Remember, it is cutting into the genome of the animal, right? So now I'm going to show you in the next slide. Now, the beauty of that is that you can easily have a transposase for the particular site that you are interested in. So now I mentioned ATP2 earlier on. ATP2 is a site that you've got transposase that would only allow you to inject things into that site. So now in this day and age, we've got companies that can do that. People in Manchester are doing that. In Cambridge, people are doing that. So they've got the resource, and they do that commercially, by the way. So they've got that transposase, and you can send your flies to them and be like, what, I want you to insert on the second chromosome. So the only thing you need to do is come up with the plasmid DNA that have got that P element. And now they can tell you, please, this type of P element is what you should use. So remember, I mentioned at P2, right? So in the type of plasmid that we use that would allow you to do at P2 is different from, let's say, something that would allow you to do different types of P element, or different type of insertion. So we use what we call the Phi-C31 integrase, I think. So that's what we use. So really, there are people that would all people inject them, but the molecular biology is a view. And note, there are companies that also can synthesize things for you. So you just need to give them, oh, I want KRAS G12V, and they're going to do that. So, but as a learning concept for students, doing the molecular biology can be fun and can be frustrating. Okay. So now you've got transposase, and then you've got your P element. The P element also is combined with what we call the helper plasmid, really. So that's how you inject it, into the blastoidal stage of embryogenesis, and then you wait. Okay. And then ideally you should have your insertion should have taken place. And then what you can do is, so this is just depicting what we've showed earlier on. So the transposase will cut, the P element will insert, and then things can segregate, and you can select, for your animal of interest. So, but you don't need to worry about this or this, because you can't do that in your lab. You really need to source it out, because it requires, special injection techniques. Yeah. Never injected. I think you've injected, and she's injected. So they can tell you, I've never injected and I'm not ready to inject. So yeah, but I don't do that in my generation anymore because, because things have changed. Oh, do you want to say something, Alima? Oh, okay, yeah. Move on quickly. So the system that we use a lot in the lab, and I think you guys will be using it as well, especially in during the course of this program, is what we call the Galfor UAS system, developed by Andre Brand and Norbert Perelman. So, and that was built from the P element concept, right? So what they are doing is that they've got what we call a tissue specific drive promoter, basically, or gene of interest, really. I'll call it gene of interest. So, and then that gene of interest is expressed specifically in maybe cell type that you're interested in or your tissue. And then you can tag that with GAL4. Sometimes the GAL4 is inserted randomly, and people, you can sequence that and identify, oh, so GAL4 is next to this particular gene, or maybe this gene can drive GAL4. So that can also, be random. So, and then UAS is never random. So you have to, do a little bit of molecular biology on what you're interested in, the gene of interest or your RNAi. You can put that, downstream of UAS, so upstream activating sequence. And the concept is that GAL4 will bind to UAS, and that will drive the expression of whatever is downstream of it. Is that okay? So, right. So now, for example, you've got flies that that have got this promoter that is specific for the wing. Is that okay? Tagged to GAL4, and then you've got a fly that will have that. So ideally, when you're working with the GAL4 US system, you want your GAL4 to be on a separate fly, population of flies, and then you want your UAS to be in a separate population of flies. They can be on different chromosomes. They can be on same chromosome. We're going to allude to that in the crossing scheme. So, right, and then your product is going to carry both. Right? So, and that product, because you've expressed, you can see that none of them have got GFP because on their own, they can't do anything. But together, you can light up the wing. Right? So, and you can quickly go onto your microscope that hopefully all of you are going to get that at some point, and you can identify, flies that have got GFP or not. So this concept is the same for anything that is downstream of UAS. So let's say, for example, you found that these 3 genes are required for, let's say, prostate cancer, and then you want to clone them and insert them into flies. You can, for example, in principle and technology that was developed in Russell's lab, you can actually put, 3 genes together. Is that okay? And express them in the wing, for example, express them in the eye, for example, express them in like 6 or 8 cell number, for example, depends on your GAL4 driver. Is that okay? So I used to work in working with group of cells that are just 6 to 8 cells, and the GAL4 driver we used then was called Snowball GAL4 because that GAL4 is only ever expressed in those 8 cells. So you can begin to play with what cell type you are interested in, right? Now, and with other systems that are similar to GAL4 UAS. So, for example, people that are interested in tumor microenvironment, you can induce something with the GAL4 because the QF and the QUAS system is independent of the GAL4 system. You can perturb the tumor microenvironment with that. And that's similar to what Ross was saying regarding, we found that this particular gene or this particular set of genes are not required in the tumor, but they're required in, for example, in the muscle. So yeah, so and another system is the LEX-AOP system. So that's another system that is independent of the GAL4. But the downside to this guy is, so the GAL4 system can be shut down using what we call GAL80. Is that okay? So the downside to this guy is that this is also dependent on the GAL80 system. So you can shut this off using GAL80. So and we can also shut GAL80 off using temperature. So now we've got what we call GAL80 temperature sensitive. So now so ideally you've got your GAL80 inhibits sorry, inhibits, not activates, inhibits GAL4, right? So that is that is the under normal condition. So at room temperature, that can happen. Even not necessarily, even so, anytime you have that GAL 80, even not necessarily at room temperature, that will happen. But once you have what we call a GAL 80 temperature sensitive, so GAL 80 TS will shut down GAL4 at low temperature, so at 18 degrees, for example. And then once you move your animal to high temperature, so even though you've got GAL 80 TS there, that won't shut down. So you still have GAL4 activation at 27 degrees, for example. So you can see that although GAL 80 can control GAL4, but we can also still control GAL 80. If you've got GAL 80 temperature sensitive as part of your system. And there were fly strains that they're just GAL 80 temperature sensitive, that even though you're working with a different GAL4 driver, you can combine them together and use them. Right? Good. So now, the concept of Drosophila crosses. So, so the reason why we actually do Drosophila crosses is because we are trying to get at some questions that with the existing fly strains that you have in the lab, you cannot get at them. So, so this is a question for us. So you keep a fly stock that carries the homozygous lethal recessive of this mutant and is wild type for the white locus on the first chromosome, and this is stock 1. This is a recessive mutant, but it is lethal. So which means if you've got ataxin 7 over ataxin 7, the animal would die. Is that okay? So that's just the meaning of that first sentence. So, and it's wild type for white. So wild type. So this is plus over plus. No, it is not white minus on the first chromosome, and that's stock 1. And you can look at the animal and okay, so this is plus over plus. Oh, it's red-eyed. Okay, that's fine. Oh, you know what? Oh, it's got ataxin 7 over curly. Oh, okay, wait. Okay, yeah, that's curly. So you know that phenotype is matching up with the genotype. So we call this genotype. So now, however, for recombination experiments, why would you want to do recombination experiment? So for example, you found that this particular gene induces sterility in animals, so prevent them from, let's say, laying eggs or even mating at all, right? And then you saw in the literature and found that actually, when people express this gene in, let's say, mouse, that actually allows them to want to mate me, for example. And you're like, and there was a strain of this, so you don't want to create your own strain. There was already a strain, but unlucky for you, that strain is also on the second chromosome. So the first question you want to ask is, wait, can I recombine that thing with this guy and see if I can rescue? So we call that rescuing of the phenotype, because you're bringing in new thing so that you can, rescue, or you can inhibit the existing phenotype. Sometimes you might want to exacerbate the phenotype, or enhance the phenotype. You can also do recombination experiment. But the reason why you're doing recombination is because this guy and the new thing that you want to bring in, they're on the same chromosome. And the next question is then, are they inserted, are they on the left arm of the chromosome or on the right arm? And we told you about the database that are available. So ataxin 7 is a type of mutation, and you already know that because you're working on that in your lab. So you know where that mutation is on the second. You'll be able to know because that is on the database. So, and now your new gene, you need to look at and say, oh, yeah, that gene, so my gene is on 2L, but the other gene is on 2R. So you can do easy recombination with that. People do recombination with things that are on 2L and 2L, but the probability of that is quite low. Of generating proper recombinant is actually quite low. So, a lot of scientists will want to use some things that, you've got, that is on different arm of the chromosome, so that the probability of actually getting your recombinant, is and there was a mathematical calculation for that, but I can't remember right now. So right, So, and then, so you want to recombine something, and you want that thing. So you want to recombine something, right? But you want your, at the end of the day, you want everything to be on a white mutant background. So what does that mean? So you want to, so you want to recombine something with this guy, but the reason why you will recombine something with it is you want the new strain to be in a white mutant background, because once you do recombination, let's say with something that's got P element, you won't be able to tell red from more red, although in some cases you can. But because this guy is already red, so what extent of red will you be able to tell, will you be able to see once you will combine something with it? So ideally for recombination, then what we do is to make sure that our strain has got white-eyed, so that once we will combine something with it, we can tell, we can be like, oh yeah, that's red because of the recombinant. Oh, that's orange because the P element is causing that recombinant. Okay. So that is why we want this to be on a white-eyed background. Now, to create a taxane that has got white-eyed background, you need to set up a crossing scheme. And the beauty, and you're lucky that in your lab, you've got what we call the balancer stock. So like I said, these are the bread and butter of what we do. So if you want to do any genetics, you really need a balancer stock. And the reason for that is they allow you to track things on the chromosome, they prevent recombination, they've got markers that you can also track. Now, so now the first question is, which I think is in the slide, is so what would be the genotype of your stock that you want to generate? And then using these two stocks, how are you going to generate them? So this is like crossing scheme 001, not even 101 yet. So, you want to be able to do that. So now, just to include you guys in what I'm going to write. So what do you think? So to answer the first question, so what do you think is going to be the genotype of our end product? So you need to know the end product so that you can walk and even walk towards it, and design a cross that will allow you to get there. So yeah, so what do you think is going to be the, no answer is the wrong answer. Yeah, please tell me. Okay, yeah, W minus over W minus. Good. Okay. Oh, thank you. Bless. Okay. So at action 7. Okay. Okay. What do you say? Oh, okay. At action 7. Yeah, good. So you remember, at action 7 is homozygous lethal, right? So which means you can have good over CYO, right? Are we happy with that? Over CYO. So why not over IF, for example? Because that's also something that is not a balancer. Good. Is that okay? So because if it's not a balancer, you can't keep a stock over if. So because at the end of the day, you're trying to generate a stock. And a stock is what you're going to keep in your lab for a while. You need to maintain them. Then you can use them for experiments out over time. So you can't keep a stock over this. Now, so how are we going to get from here to here? Right? So now, so can someone tell me what would be the first cross that we need to do? First cross. Yeah, please go on. Yeah, please go on. Good. So that is the third chromosome. So that is telling us that the third chromosome is also wild. So there is nothing on it. So yeah, so that is just showing the third chromosome. Yeah, good question. The question is that what would be our first cross or first animal that that we need? What male or female do we need in the okay? So yes, so for me, because I know that I know my end product, right? So the first thing I'm gonna do here is, and because we want to make sure that we've got white minus over white minus, that's the goal. That's the main goal, right? So we need to dilute the plus over plus. That's the first thing. And how would you dilute that? So note, this W over W is for X over X. That's for female mainly. The male are going to be hemizygous, so that's going to be W over Y, right? So if you're going to use male of this guy, you won't be able to dilute this properly because some of them would have, and some of them will have Y. Is that okay? So the first thing to do is to use female from here because you've got these two copy of white minus, and you can use that to dilute this guy properly. So that's the first thing. Is that okay? So you want to dilute out your plus. Now, how would you do that? We know that now that the answer is already out there that you need female from here and you need males from there, right? So now we're going to write that out. So we need males from the ataxin thing. So males, so that's going to be plus over plus. So I'm going to shorten ataxin to ATX over curly, right? And then so we do need to write the third chromosome because that's wild type. Is happy. So now we cross that to a virgin female. Note, you must use virgin females anytime you're setting up crosses. So, and that is W over W. And this is if over curly. Right? So that's if over curly. Now, based on Drosophila crosses, the homologous chromosomes are going to separate, right? And based on genetics as well, homologous chromosomes are going to separate. Now, and then you're going to have mixed match, right? So males go to female, female goes to male. Now, what you can have here, the set of genotypes that you can have here includes plus. So from the first chromosome here, you match that to this, and you've got males and females for that. From here, you match that to this. From here, you do that, and you do that. So, and how would you do that? You need to consider the chromosomes as well. They are homologous, but they're separate. They're independent, right? Mendelian's law. Is that okay? So independent assortment. Is that okay? Segregation. So the homologs are going to segregate, but the chromosomes themselves, they're separate. They're independent of each other, right? So what you just need to do is you've got your plus over W, right? Because these guys are independent, so you're going to have one of ataxin, let's say, for example, so this is arrow, over, let's say, if, for example. So those are females animal, right? Now you're going to have males as well, right? So plus, so because you've got males here, and oh, I take it back. So sorry. So the males are actually plus over Y, right? So the males are plus over Y, right? So you've got plus over W. Oh, thank you. Yeah, I think it's moving for some reason. I don't say if that one. Yeah, I think it's moving. Yeah, it is moving for some reason. I hope it doesn't though. Right, thank you, Ross. So so you've got the females, right? Now we need to look at the males for that. So that would be, let's say, Y over W, right? And then you've got your semicolon. Say if again, because that is independent. That second chromosome is independent of the first. So you're going to have these two, right? So now the next thing we can have here, so So this is 1, 2. So the other genotype we can have is what? We can have plus, okay, yeah? Okay, good. Yeah. So, and we can have the males and female, right? Okay, good. Yeah. So what else are we gonna add? Sorry. Yes, you can have CYO over CYO, but that that's gonna be lethal, right? So another thing that you can have is this if over this curly. Is that okay? So you can dilute out this. So number 5. So you can have plus over and then if over curly, right? And then the next thing you can have, good. So that would be the male, right? 6Y, right? So now, now remember we've listed the genotype. So how can we tell what exactly the phenotype? Right? So you know the genotypes are going to come out of the crosses, but you need to be able to select the right one, right? So now the phenotype of this guy, they're going to be rough, they're going to be the irregular faceted, they're not rough eye, the irregular faceted, or facets actually, irregular facet, phenotype. So that will be the phenotype of this, but they are still going to be red-eyed because they've only got a copy of the white minors, right? Now the males, number 2, that's also going to have the marker, the IF marker, but they are going to be white-eyed, right? Because the white minus is already there. Like, and Y doesn't really, Y is useless for some reason. I don't know why. But Y is useless. So this is going to be, a white-eyed animal, but with the irregular facets marker. And then number 3, you have your wild type animal, wild type eye, red-eyed, and then that's gonna be curly. So you don't want it because that also came from your original experiment. So you really don't want that. So and then number 4, that you're gonna have Y over W and then ataxin over curly. So that's also gonna be white-eyed but curly. And then number 6, we've got white-eyed and then if over curly. They've got the 2 markers, so you don't need that. And number 5 also, I've got the 2 markers, you don't need that. Now, the next question is, so are we going to use number 2 or number 4, and why? So what do you think for the next stage of the crossing scheme? So do you want to use number 2 or number 4, and why?
Because number 4 has
Good. Good. And what sex of the animal are you going to use? Remember, this is not the end of the male. Good. Now, because it is CYO, very good point. What are you going to cross it with? You're going to cross it with the balancer, right? So, and will you be able to tell the offspring? Because if you're only going to be selecting things that have got CYO, now at some point you're going to run out of CYO because you won't be able to tell, oh, is this my parent or is this the offspring? So what else do you think we should use? Remember, I said the combination takes place mainly in the female. And the reason why we use the markers is because we want to be able to track genetics, right? In addition, we've got balances because they can recombine. Now, the male here has got IF, sorry. And then we know that the combination won't happen. So, but for us to be able to create a stock, because the next actually, the next stage of the cross is actually going to look at you're going to develop your stock from there. And I'm going to show you. So, but here you've already developed like one part of your stock, because you can easily, this white-eyed, you're fine with it, but they're only males, right? So you need a female in the system. So that female needs to be over curly. So, and for you to be able to track that curly, the males needs to be Eve. Right? So that's the only caveat that you need to bear in mind, that, what am I trying to look at and what's my end goal? Your end goal is that you don't want to have Eve in the system. So you can pick males from these, because the one we combine, they've got that phenotype, and then you can cross them to your balancer stock, and at the end of the day, you can pick out what you need. Exactly. Because from there, you will be able to pick out both male and female, and you can cross them together. Now, so the next stage of this cross, so because we're going to select this guy, right? So, and then now we're going to cross that guy. So plus, oh, sorry, y over that. So y over w, atx over Eve. So now, so out of these two stocks, what are we going to cross to? Stock 1 or 2? Stock 2, right? Because we are trying to dilute out our whites, and we've already done one, so we need to reuse it again. So, right, so we use males here, and then females here. So remember, virgin females. So, and Eve over curly, right? Now, your offsprings are going to be similar to what we've done here, the laws of Mendel's, right? So we've got y over the first W, and then ataxin over Eve, right? And then the female of that is going to be W over W, ataxin over Eve, right? So 1, 2. And then 3 is going to be what? So Y over W, I can hear that. And what? Ataxin over CY. And then 4. Okay, good. And then 5. Y over W. If over lovely. And then 6. Okay. Thank you so much. Right? So now, these are the genotypes, right? So, and then, so what phenotypes do we want? So the number 1, that's got Eve, right? And that's a male. That's still useful, right? It's still useful, right? So still useful. So number 2, that is white eye, but it is ataxin over Eve. So that is useless because it's got the marker, not the balance, and it's a female, right? So the recombination will happen. Now, number 3, we've got Y over W, ataxin over curly. So that is a white-eyed male, and it is over curly. It's still useful, right? Good. And number 4 is W over W, ataxin over curly, right? So, and is that white-eyed or red-eyed? So number 4, good, it's going to be white-eyed, right? And that is very, very, very useful, right? And then number 5, we've got the markers and the balancing. So we don't really need them. So they're useless. Now, we've got something that is likely to be useful and something that we think is also likely to be useful. So 1 and 3. So, and they're both males, they're white-eyed, but they've got different markers, really. So which one are you going to pick? 1 or 3, and why? 1 or 3, and why? So for those that cannot see, so 1 is Y over W, ataxin over Eve, and then 3 is Y over W, ataxin over curly. Why? Good. Right? So we pick these because now, anytime you cross these two, because now you're going to be creating your stock. So you're at the end of your crossing scheme because you've achieved your aim of diluting out that plus. Is that okay? So now you've got your Y, W over W, you're happy with that. You know, those are females that you're interested in, that's fine. You keep them separately. And then you've got your Y over W, ataxin over curly. That's also a Y type, and it's got ataxin and it's got curly. You're happy with that. So you're going to cross them together and you're going to create an homogeneous stocks from them. Homogeneous stock from them. Is that okay? Now, let's say you use ataxin over Eve. You're still gonna have some Eve over curly once you cross those together at some point, and you know you're gonna continue the cross to non-stop. So to actually stop the cross, you are happy with these two stocks or these two generations, these two offspring set from them, and then you can create your own stock. And now, so we call that. So I'm not sure if this is French, but my PhD supervisor used to say interse. So I'm not sure what that is. I've actually not checked. So interse means, you know, these are the same genotype and the phenotype, and you're just crossing them together. Okay. So here, to create a stock, you're just going to have ataxin over curly cross to these over this. And that gives you this. So, and that's your stock. So any experiment you want to do now, you can you can do it. So you can do your recombination because that is on a white-eyed background. So happy with that? Any question? Question? So right, so this is quite an easy one. So you add about red today, right? So, oh, thank you. So medullary thyroid carcinoma, thank you, right? So, and now you want to, generate your own cancer model from this. So this is a point. So now, so the question, so the thing is, so you want to test that if a kinase called RET, so RET is a kinase, is a tyrosine kinase, so is oncogenic in Drosophila. So, and you decided to express RET in the fly eye. Some people have done that. And you have got two strains. So strain 1, your driver, and strain 2 that has got red, right? So now, a little bit of detail about this genotype is that, so you've got your red-eyed animal. So that shows you that, that is plus over plus, because it is not white. Thank you so much, Aline. So it is actually plus over plus. And then you've got your driver, GMR. So GMR, we said, the Galfa US system requires what kind of answer. So here, GMR is an answer that can allow Galfa to be driven to the eye of the animal. Is that okay? And Ross can give you really comprehensive detail about GMR and everything. I'm not gonna do that. So GMR is a Galfa. It is over curly, so that's a balancer. Thank you. And then you've got third chromosome that hasn't got anything on it. So it is wild type, right? Now, there was another animal set or another strain that is also, wild type for the first chromosome. It's got what we call UAS, Drosophila ret, and with that point mutation. You can also, for example, according to what Ross said, you can also use human ret, and that's been done. So, and we can use Drosophila ret as well. So we can have UAS, H human, ret, let's say S something T. Okay. And then over curly plus over plus. So, and actually next slide will be good for that. So yeah, so plus over plus. Now the question is, what will be your genotype? What genotype will you look at for the experiment? Because you've got your question that you're trying to get at. And then how will you design, a crossing scheme for this? So this is one of the, the easiest crossing scheme that you could design, right? So So how would you, so yeah, so what would be your, the genotype of the strains that you want to look at, or of the strain that you want to look at? Oh, hmm, any answer, any taker? Say some. Yeah, so what would be the genotype of the strain that you want to look at? So you're trying to ask if this guy is oncogenic in Drosophila. And the lecture this morning actually alluded to a little bit. So what genotype are you gonna look at using these two stocks? You know the answer, I know that. The second one. Good. So now the second one has got, yeah, you're right, you're gonna use this, but what will be your final genotype of that strain that will allow you to test if ret is oncogenic in flies, right? So now you need a combination of these two strains, right? So your final strain is actually going to be GMR. So I can write plus over plus and leave the third GMR gal4 over UAS direct. Now you can add the mutations if you want. So that would be your final stock. So and that stock, sorry, and that strain, not stock, will allow you to be able to be like, okay, so because we need a GMR drive something to the eye of the animal, what happens to the phenotype of the eye once you express this strain or this mutation? Now, okay, so you can quantify, the eye is rough, oh, the eye is lost, things like that. So and that's the genotype. Sorry, and that's the genotype of what you're gonna. So that's your experimental animal. So ideally, you don't keep a stock of this. And the reason why you can't keep a stock of this is because you can't even keep the female because of the recombination. So, and we do recombination with this type of things as well in the lab. So we combine maybe a GAL4 driver, let's say with GFP. So we can use UAS GFP and we can select for animals that are GFP positive because we know that the GAL4 is there and we can use that for other experiments. Okay. So, but for this oncogenic assay, you don't do that. And a lot of the time, even the animals won't survive. So yeah, so you keep the stock separately, and then you keep the GALPA separate from the UAS. And once you're ready for your experiment, let's say you want to test the effects of the compound, you cross them together, and then this progeny is what you're going to be looking. Right. So, and how will you design, a crossing scheme for that? This is quite straightforward because what you can do is you can use either male or female. And why do you think you can use either male or female? I can see some answers because correct. So because you only need just one GAL4 and one UAS. And note, because they're both on the second. So let's say one of them is on the first, then the story changes, right? So, but because they're both on the second, you can either use either male or female. So ideally what I do is, you can use, let's say, female of your GMR GAL4. So that's going to be plus over plus GMR GAL4 over curly. That's the one. Is that okay? And then cross that to a male. So that's plus over Y. UAS red, sorry. Over curly, right? And then the offsprings are going to be, so you've got plus over plus GMR over red first, right? And then you're going to have male of that, I think. Right? So that's 1, 2. So 3. So what are the genotypes? So we can also have plus over plus, GMR girl 4 over curly. And then 4 plus over Y, GMR girl 4 over curly. And then 5, right? So we can also have curly over US red, for example. So we've got plus over plus, curly over UAS red. And do you know that plus over plus curly over US red is the same as plus over plus US red over curly, right? Happy with that. So 6 half plus over Y, plus over Y, curly over US red. So right, so now, so you can see that although you set up a cross to look at one genotype, you can see that you've got multiple genotypes. Now, from here, because of the way you set up that cross, you know that anything that I've got a balance, you're not interested in it. Right? So the genotypes that you're interested in are the plus over plus GMR gal 4 over US red, that are females, and then the plus over Y GMR gal 4 over US red are males. Okay. And anything that I've got a balancer, bin. Happy with that? Good. Now, and the last part before we maybe wrap up for the day is not this. Yeah, this. And this is, this is actually based off of, people that are computation, people that are interested in, in just asking questions. And, you're wondering, wait, how do we go from, oh yeah, go on, please. Sorry about that. So yeah, so people that are interested in like asking questions and, you know, trying to get at questions using Drosophila, and you've got something that you want to test, like an hypothesis or something, and and you're like, wait, how do we go from from just looking at something from a database into actually testing it in the lab, right? So, and here I'm going to use KRAS as an example because I work on colorectal cancer in the lab. Right. So Ross actually alluded to that. KRAS is really the one of the most scary thing about cancer because it's mutated literally in almost half of cancers. Really difficult to drug. Drugs are available. They're giving patient really minimal effect. Most of these drugs are not working for colorectal cancer patients. So KRAS is something that a lot of people studies it. Like a lot of people studies it, and people study it from genetics, structural biology, computational biology, from cells, from flies, from mouse and things like that. So it's really the body of knowledge regarding KRAS. So from your literature review, or your computational studies, you found that KRAS is a gene that is recurrent in patient basically. And then you want to model this in flies, right? So now we've got several questions for you. So one question is, how will you go about it? Where do you actually start from? So what technology are you going to use? What fly strains do you need? And how are you going to generate them? Design your crossing schemes. What readout are you going to be looking at? And what questions you want to ask after that first experiment? Remember, everything you're doing from here to here is about just trying to do one experiment. So, so how would that inform the next experiment? And then how do you determine, is this impactful? Have I even contributed to knowledge? Okay, so, and we're talking about patents, talking about publications, right? So now, It's gonna be a bit interactive and can I can like thank you. So right, so the first question, so is that how will you even start thinking about KRAS? You stumble upon this gene and you're like, you know what, this gene is actually recurrent in patients. What the first thing you what would be the first thing? I've got my way of approaching it, so I really want to know what would be the first thing you're gonna do. Right? Anyone? Anyone? DRTC even? Anyone from DRTC? Yeah. So what would be the first thing you're gonna do? No, Festus is gone. Right. So to me, and you can you can have different approaches to this because you've done your literature review, right? And but you just find out this guy is recurrent. So the first thing you do to me is that you try and identify, wait, what type of gene is this guy? Because you don't know what that mutation is doing, right? So you want to ask, is this an oncogenic mutation, or is it a mutation that leads to the loss of function of that gene or of that protein, loss of function of protein or gene? Okay. So so that's the first question you want to ask. So what type of mutation is this? Because that's the only way for you to model it in flies. If you don't know the effect of that mutation, then we're wasting our time. Okay. So that's the first thing. So now, you went into the literature and you read and you're like, wait, so KRAS is a proto-oncogene. So a lot of mutation that happen here, they should be oncogenic. You have to check that in the literature and make sure that you find that any mutation in G12V is actually oncogenic for KRAS. So now you know that is oncogenic. And what is the meaning of oncogenic, right? So the concept of cancer is that you've got activation of proto-oncogenes and the loss of tumor suppressors. That's one of the definitions, right? So now what you're going to be thinking of is that, okay, yeah, so there is activation of anything that is downstream of this particular guy. So that this guy is, for example, being activated. Is that okay? So another concept of that is that, oh, there is a law of this thing, or a lot of that pathway. Is that okay? Been taking place in those cell types. So you've answered your question by going into the literature and you're like, here, G12V mutation will lead to activation of this thing. Now you need to think about, so it's how to model this in flies. So what technology am I going to use? Right? Okay, this is an activating mutation. I'm looking at KRAS. Flies don't have KRAS specific gene, that is human. Human KRAS is not in flies. They've got their own autolog that is RAS, but they don't have KRAS. So now you're going to be like, wait, do I want to use a RAS that is the autolog in flies, or do I want to shuttle the human mutation into flies? You do that. That's part of it. That's part of the thinking. Yeah, you have to compare both. Yeah, are we ready for? Yeah, I'm happy to leave, and then we can continue from here. Is that okay? Because I'm starving as well. So, and this is a good place to stop. And no, please think about it, and we can continue. Right. So thank you so much, guys.